Okay, let's get started. By my watch, it's 5 p.m. Singapore time on Wednesday, 4th November. A very warm welcome to everyone joining us on Zoom. On behalf of the SingHealth Duke-NUS Global Health Institute, I'm delighted to welcome all of you to this final session of our three-part webinar series on healthcare. This series focuses on healthcare innovations in response to COVID-19. As some of you might know, the Institute aims to promote research, education, and capacity building initiatives in the region. And we're very pleased to form this community of learners that has been gathering to all our webinars and other seminars. And we try to bring you a whole range of relevant topics. In the previous two webinars of this series, we brought you examples of innovations that were based in hospitals and in clinics, as well as the community. In this webinar, we provide an overview of some of the exciting research innovations taking place here in Singapore, but which will be very relevant in the region and in fact the whole world. So we're really looking forward to the discussion today and hearing from our esteemed panelists, Dr. Linfa Wang and Dr. Dean Ho. I'd like to now introduce our moderator for today's session, Professor Gavin Smith. Gavin is a professor in emerging infectious disease programs at Duke-NUS Medical School and is also research lead for the Global Health Institute. He is primarily interested in the roles played by mutation, natural selection, recombination, and resourcement and host immune response on virus diversity within an individual, during transmission within a population, and during interspecies transmission between hosts. While he works mostly on influenza, he also studies a wide range of respiratory and enteric viruses. His research program is directed at efforts to better understand viral disease ecosystems in Asia, especially the animal-human interface to inform and enhance disease control. In the context of COVID-19, he has engaged in research on genetic changes in the SARS-CoV-2 virus and their potential implications for the course of the COVID-19 pandemic. Bringing this wealth of knowledge to this seminar, we are delighted to have him moderate this session, and I'd like to thank him again for taking the time to be with us today. Before I hand over to him, please note that the webinar is being recorded. Please post your questions in the question and answer box at the end of the session or anytime during it, and they'll be addressed at the end. And please keep yourself on mute. And now over to you, Gavin.
Thank you very much, Amina. So welcome everybody. It's a pleasure to host you today for this seminar that is looking at research innovations in healthcare from the Asian region to test for, prevent and treat COVID-19. We have two very accomplished speakers today. Just before I introduce them, a little bit about the format. So each of the speakers will be given a short 10 to 15 minute presentation. And then after that, we will have a panel discussion. We've received some questions in advance, but as Amina mentioned, please put your questions into the Q&A and then we can ask them of the speakers. So the two speakers, first up will be Professor Linfa Wang. Linfa has been in the program in emerging infectious diseases at Duke-NUS Medical School for a number of years, and I've worked very closely with him during that period. Professor Wang is an expert in the field of zoonotic diseases. He has been very active in the response to COVID-19 in Singapore and globally. His research interests include zoonotic diseases, bat immunology and pathogen discovery. And his research has extended from looking at bat-borne viruses to try and better understand virus-bat interactions and how the bats can coexist with a large number of viruses without actually getting sick themselves. And this is, of course, a key point to understanding disease emergence. In terms of the COVID-19 response, Professor Wang has made significant research contributions, including developing an antibody-based serological test to detect SARS-CoV-2 virus, and also was involved in the early and successful culture of the virus from an infected patient sample here in Singapore. And his team has also been working together with local and international partners to develop new vaccine and therapeutics for COVID-19. And if he wasn't already busy enough, he is also involved in multiple World Health Organization committees on COVID-19, and at the same time is contributing to important publications in this area. If you're ready, Linfa, we can go ahead, and then I will provide a introduction for Professor Ho directly before his talk.
Thank you, Gavin, and I'm ready. So you can see my slides good?
Yes, all
okay.
Good. Thank you.
All right. So yes, it's always a pleasure to speak in this forum for the ASDG-HI. So today, I think I'll do a slight differently, right? So we are going to just very briefly describe the science we have been doing, but really focus on the impact. So I'm choosing a title, combating COVID-19 from bench to bedside, but / field side, because it's not only bedside, there are still questions remaining in the field side as well. So what I'd like to start with is that, in July, Nature really published its commentary to say 6 months, from January to July, 6 months later, we're still facing the mysteries that scientists are still sort of trying to understand. So these are the 5 Nature sort of highlight key unanswered questions. Why do people respond so differently? Means some people get no symptom, other people die. What's the nature of immunity and how long does it last? The antibody versus the T cells. Has the virus developed any worrying mutations? How well will a vaccine work? And what's the origin of the virus? So when I was prepared for this talk, I thought, actually in Singapore, we are doing pretty good because we have made progress in each of these 5 areas. So that's what I'm going to, briefly describe. And I believe the lucky number is 6, so I'm going to add in 1 more. So I'm going to give you the 6 examples of our contribution from Singapore. And this can be done in chronological order. So end of January, as Gavin already says, the team led by Dr. Daniel Anderson was the first one to isolate a live virus, the second country outside of China. That laid the foundation for all the subsequent work we did, whether it's diagnostic, vaccine, or animal models. Then end of February, so 1 month later, we were the first to apply serology in contact tracing. I have been in the field for 30 years, and maybe I was joking, I'm old-fashioned. I always a strong believer that, the modern genetics, next-gen sequencing is important, but serology still has a very important role to play. So from day 1, when I heard of the new pneumonia caused by novel coronavirus, I was already planning and working on 6 different antibody tests. So months later, we applied this, and this is, I think, for those of you in Singapore, you knew that initially we have 4 clusters of important, COVID-19 cases, and then these 2 clusters, we found the link between these 2, and these 2, we also found the link. But then among the others, these 2 major clusters, we believe there must be a link, but we just could not find it. So unfortunately, in the acute phase, we missed that. And then a month later, we finally traced down a couple and using antibody tests. By then, the virus is already cleared. So we were able to trace that link. So that's the first application of serology. Now, March, 1 month later, the study led by Gavin Smith, who is chairing this session, we discovered a major deletion mutant. Up until then, the SARS coronavirus 2 has been really considered as genetically very stable. After millions of infection, you have a few point mutation, but you don't have major genetic change. So Gavin's team was the first to discover that major deletion of 382 nucleotide. And that led to a collaboration with the clinicians and immunologists to further define to say that patients infected with this mutation actually suffers less severe disease. So that's very important to understand disease severity. And then May 20, we launched this antibody test Gavin already mentioned. Its commercial name is CPAS. The difference is that it measures the function neutralizing antibody, and I'll come back to this later. July, we made a discovery led by the team of Professor Antonia Battaletti, also in our department. This is another very high level of basic studies, but have a direct impact on the standard disease. Basically, Antonio demonstrated that the T cell immunity is very important, not in terms of preventing infection, but it's important in terms of prevent diseases. So this was cited even by the director of the NIH in USA, so it's a very high profile when it came out. And then August 2020, the team led by Professor Wang and Dr. Jenny Low put this vaccine co-developed with the Singapore scientists and Arcturus into a clinical trial. So this is obviously, I mean, we always say for vaccine, there's such a fast pace. We call there's front runners, but we also have the next generation vaccines. We believe that there will be more advanced vaccines. And then this one, the Luna 19 is in that category because it uses a replicating mRNA. So it's a different, slightly different from the Moderna vaccine. So now I'm going to just go to the, give one specific example of my own research on how serology can play a role in Singapore, regionally, and globally. So the challenges for COVID-19 in terms of understanding serology, personally, I think these are the list. We talk about herd immunity, the immune passport, how long the protect immunity will last. Can we use convalescent plasma for therapy? And the vaccine, of course. Can we have a safe vaccine? If we have one, how efficacious and how long the immunity will last? And then the last one is the Nature in the paper also identify the origin of the virus and the role of intermediate animal is still unknown. So I classified this. The first 5 are human related in red, and the last one is animal. For the human related, basically, that we need to measure the functional neutralizing antibodies rather just a binding antibodies. For the last point, for animal works, then we need what we call a species independent serology. We want a one-for-all test. We want a test we can measure neutralizing antibody to the SARS-CoV-2 or closely related virus, not only in humans, but in any of the animals from bats, pangolins, to civets, to pigs, you name it. So we invented this technology called a surrogate virus neutralization, really address both of these issues. This is the test now we can measure neutralizing antibody in a species independent manner. And as I said, I'm not going to science too much. This is already published. And I just want to illustrate, in our EID field, I always say it's almost like army. We have peacetime and wartime. And we need to work really, really fast. And for COVID-19, this CPAS is one good example. We had this idea in early March, and we discussed the invention with our commercial team. And then we got the data, patent, commercialization, licensing, approval by HSA in Singapore, the equivalent FDA, and we launched commercial kit in May 15. All of this was done in 70 days. So that's really is necessary if you want to be in the game. So what I'm going to give you examples of not our study, but studies from international teams using our test. So this is really a very interesting study is conducted by a US scientist in Seattle, Washington State. So they monitor a team of workers that we went on a fishery sort of out trip to the sea. And before they went, it's total of 144 people, 122 individuals. And they measure their really base level by PCR to make sure they don't have active infection, and by antibody test to make sure the baseline level is known. So they used the 2 antibody tests, the lab-based. One is the Abbott test. So this is the high throughput test commercially. And another commercial test is the ACE2 binding block, which is a CPAS. So as you can see, the Abbott test identifies 6 individuals out of 122 as antibody positive, whereas our test says only 3 of them have real neutralizing antibodies. So that's the difference to start with. There's a 50% difference in the core of what considered as a positive antibody response. But the real important is that then in the next 10, 20, 30, 40, 50 days, some of these workers will come on shore, and anybody coming back, they also did antibody tests and also PCR tests. Now, what they found is 85% of these people went to the sea coming back with positive. So the attack rate is incredible, 85%. And yet, these 3 individuals who had these neutralizing antibodies were all negative when they come back, and then their antibody dropped a little bit. So I think this is a very good illustration of this test can measure functional antibody. Also, this study demonstrated that if you have a high level of neutralizing antibody, have an inhibition in our test, 80% above, most likely you will be protected. Okay, so then I will finish up with another application. Obviously, we are working very hard is hunt for the origin of SARS-CoV-2. And this is a commentary come out of The Economist and cited with two very high important sort of key opinion leaders in our field. Peter Daszak is the CEO of EcoHealth, and Jeremy Farrar is the CEO of Wellcome Trust. Both of these discipline scientists basically think, the SARS-CoV-2, the origin of this may not be limited inside China. They made this conclusion is coming from this distribution of bats. So as you can see, this is Yunnan province, southern China, the most close related, and Wuhan is somewhere in the middle. So the most close related virus in bats is discovered in there, 96%. But if you look at the bat diversity and bat density, the neighboring countries in Myanmar, Laos, Vietnam, all the way to where we live, the Southeast Asia basically is really the hot zone. So this is where we're going to using our CPAS assay, because as I say, it measures neutralizing antibody much more specific and in an animal independent way. And that's why we're going to do this. And luckily, we recently got 2 WHO fundings. These are very sort of urgent calls. So one, we're going to focus just on Southeast Asia, mainly collaborate with scientists in Thailand. And the other one is to collaborate with EcoHealth. And we're going to go beyond, Asia, go to, Middle East, South America, and Africa. So this is going to be really try to use our test, really go beyond Singapore, the region, and try to make an impact globally. And with that, I'd like to thank, this one Singapore approach. We talk about one house, but also one Singapore, because I mean, Duke-NUS, many of the scientists are leading, but without this collaboration with the other institutes, we will not be able to make this kind of impact. And with that, I'd like to thank you for your attention.
Thank you very much, Linfa, for the overview of the activities and the usefulness of the CPAS. Just for everybody's information, we'll have the Q&A session at the end. So right now we'll move on to the second presentation by Professor Dean Ho. Professor Ho is a Provost Chair Professor and Director of the N1 Institute for Health, as well as being the Director of the Institute of Digital Medicine and the head of the Department of Biomedical Engineering at the National University of Singapore. Professor Ho is very active in the area of applying artificial intelligence to healthcare, and with Curate AI platform that has been developed, has led multiple pioneering clinical studies in personalized medicine. And there are many clinical studies that are ongoing or have started in areas of solid cancer, blood cancer therapy, digital therapeutics, and post-organ transplant immunosuppression, among others. Professor Ho is being well recognized for his contributions and achievements, and has been elected a member of the US National Academy of Inventors. He's also received numerous awards, including National Science Foundation Career Award and Wallace Coulter Foundation Translational Research Award, again, among many others. So thank you very much, Professor Ho, and if you could please share your screen and begin. Thank you.
Great. Thank you so much. First off, I wanted to thank Amina and team at the SDGHI for the kind invitation to be here today. It's also wonderful to be here with Prof Wang and Prof Gavin and discussing kind of the work that we're doing to harness AI and digital medicine to advance drug development. So looking at the interventional side of potentially identifying combination regimens quickly that could be of use, certainly for evaluating emerging pathogens downstream. So I wanted to start off by showing this figure. So we had published a paper maybe about 8 months ago. It's entitled Artificial Intelligence for Cancer Therapy, but in the context of AI in drug development in general, it's certainly applicable to many other indications, including infectious diseases. And when we look at this roadmap here, A lot of terms are often used interchangeably. Of note, when we think about AI and therapeutics, there's the concept of discovery, identifying targets, lead generation and optimization, as well as development. And in AI, these terms are often used interchangeably, but they're actually very different segments of this roadmap. And both certainly need to be addressed and seamlessly integrated if we're ever to optimize regimens for all kinds of indications. The challenge is when we think about COVID-19, we don't have a lot of time, right? And so probably one of the more go-to methods has been drug repurposing. The notion of drug repurposing is important. It's been important for many different diseases out there, but the concept of just pinpointing compounds in drugs, that's one important element of developing an optimal regimen, but there's a lot more work that has to be done. And in particular, during the development stage, optimizing combination therapy design is important. And so to do what we do, we apply this principle known as we are all parabolas, or the second order interaction between drugs and doses as inputs and efficacy and safety as outputs. And through a neural network derived and based approach of pinpointing this relationship, we are able to achieve optimization through a minimal number of experiments. And this is something that we've applied both from the in vitro through preclinical all the way through clinical stage of drug development that we thought could potentially also be applicable towards COVID-19. And here's just some examples. This is a patient that we treated with prostate cancer. We found a much lower dose that actually increased his efficacy. And we've addressed this for other indications as well, all the way to HIV, TB, and beyond. But the core notion here is with this relationship, we can drastically reduce the number of experiments needed for optimization. Now, if we think about AI in drug development, it's all about a massive drug in dose space that we have to reconcile quickly in order to really optimize care. So I'm gonna do this quick animation here, where in traditional combination therapy development, there's this concept of, maybe drugs 1 and 2 together are the best. And in the very early stages of repurposing against COVID, when it wasn't even called COVID-19 at the time, combinations like Kaletra were proposed very early as potential candidates, and subsequent clinical trials have shown potentially otherwise, right? And there could be 3 drug combinations that are good. There could be 4 drug combinations that work, but in traditional drug development, the combinations are picked first, right? So it's kind of a one-shot deal to see if something's going to work. And we get approached often. If we've got a lot of data, can we help people find optimal combinations? And using a traditional AI-based approach, there's a lot of pre-existing data, but the issue is combinations are often given in fixed dose, right? You can have 5 million data points, but if combinations are given in fixed dose, that's in fact a very limited range of information that's provided, even though you have a lot of data. And so we have this important concept where it's not necessarily the amount of data that you have, it's actually how you acquire the data that can be the biggest differentiator in truly optimizing outcomes. And with traditional AI, using a lot of this data, but at fixed dose, the outcome is generally going to be, hey, out of these 3 regimens, can you find us the best regimen that's broadly speaking good for a population? So this is number 1 out of 3. What we do is quite a bit different, all right? So if we're going to develop a combination, why don't we go ahead and increase the number of drugs that we consider, all right? But here's the challenge. If you take 12 drugs, and 12 is not a very big number, and knowing that dosing matters too, and you try each drug at 10 different doses, that's actually 1 trillion possible drug combinations. And the resources just don't exist to be able to crunch through this prospectively and experimentally. Not only that, let's pretend drug 5 doesn't work on its own, all right? Let's say drug 5 applied to whatever disease model, liver cancer, coronavirus, et cetera. Let's say drug 5 is ineffective. It's often removed when we think about traditional drug screening. We don't remove it because, again, aside from mechanism of action, when we put drugs together, oftentimes it's unexpected drug interactions that really mediate globally optimal outcomes. So instead of combing through old data. We don't use any old data. We don't use any in silico modeling either. We use far fewer data points, but we acquire our own data prospectively by exposing, for example, the live virus to very strategically designed permutations of different drugs at different doses to outline the boundaries, essentially the galaxy of possibilities from which we apply our second order response, and then we identify the best possible combination from that. Our experiments don't yield one answer, right? Our experiments yield a ranked list of thousands of combinations from best all the way to worst based on actual experimentation. And this is a process that we apply in a rolling manner from in vitro to in vivo, where we re-optimize dosing all the way to clinical in order to re-optimize at each step of the way to make sure that whatever is pinpointed in the in vitro stage will hold all the way through, right? It's not just about finding something that's synergistic here and hoping that it works here, because in fact, that's how a lot of the drugs fail. And we tell people, we work with many drug companies, trial failures certainly happen in late stage, phase 1, 2, 3, but oftentimes the real failure happens here in really what that initial lead candidate combination was. And so not long ago, we applied this concept to the coronavirus by running Identify, this platform, to pinpoint combinations off of the live virus using, at the time, we started this many months ago, and working with the CDC, a former epidemic intelligence officer, we pinpointed 12 drugs that we thought would be the most actively used in clinical trials around the world. And we designed our experiments, and from these experiments, we looked at, and again, we were actually running a new experiment with newer drugs, but at the time, this included the remdesivirs, the favipiravir, hydroxychloroquines, et cetera. And we got asked a lot of questions right off the bat. You know, why hydroxychloroquine? Why Kaletra? And we ran these as a benchmark to see if our assay would align with many of the clinical results that were either underway or had already completed. We didn't want to apply 12 nuclear warheads at this. We needed some calibration to ensure that this was accurate. And again, we don't use any pre-existing data. We actually run experiments. And by the way, a lot of the pre-existing data doesn't exist. Right? Out of these 12 drugs, we need very specific drug dose permutations to stretch the parameter space to make sure we are indeed interrogating that galaxy of possibilities. And that's why we run our own experiments. We don't do any in silico, right? And surprisingly for us, when the clinical communities approached us, the first questions they asked were not about what the top combos were. The questions they asked were, can you tell us how Kaletra fared? Can you tell us how hydroxychloroquine and azithromycin fared, to see if it aligned with those clinical outcomes? And knowing that we don't use any of that clinical data to do this optimization, indeed, right? The hydroxychloroquines and the azithromycins were relatively ineffective. Kaletra was relatively ineffective. But through this agnostic approach, we did see that the remdesivirs plus the Kaletras yielded a very interesting outcome in that unexpected drug interactions emerged at clinically relevant doses to mediate what were very huge increases in inhibition, right? And the thing is, we've done this for oncology, we've done this for all kinds of indications and moved them all the way to clinical, substantially improved clinical outcomes, while demonstrating that the in vitro optimized combination held true all the way through to the clinic. And the reason for that is because instead of finding the best combo out of 3 options, out of 1 option, out of a few options, we are finding the best option out of hundreds of thousands of possible permutations, or even 1 trillion possible permutations. Again, clinical trials really do fail at the very early stages when suboptimal regimens are developed and seemingly work well preclinically, right? This is about running a minimal number of experiments to find true optimums. And so again, beyond just 1 option, we yield many. And so this is just a small table representing where the different regimens show up on these rankings. And again, a lot of we see this every time. Cancer and beyond, drugs or combos that work well or that do not work on their own oftentimes end up in optimal combinations when delivered with other drugs, but at the right dosage and combinatorial design permutations, right? And so we can see one major conclusion is that we see that, you know, remdesivir alone is interesting, but in properly designed combinations, it might do a lot better. And so with that, we're going to run with much newer drugs moving forward, but that work I just reported is in press. It'll come out in a paper pretty soon. And again, it's agnostic. You know, if someone were to come to us and say, look, we need only locally available drugs that are accessible to the community, and we need options quickly, we can completely identify experimentation in about 2 weeks or less, give or take, depending on the number of drugs and assays we want to run. Final thing is, for a little bit of fun, you know, our team is beyond engineers and AI and clinical collaborators and et cetera. We brought together a team to make a comic book called Aiden and Eileen that clinicians, engineers, behavioral scientists, healthcare economics researchers, free for download, to kind of teach future leaders in innovation, our early learning community, about how AI can impact infectious diseases. And we've got other books in the series looking at other diseases as well, but feel free to have a good time with that. And then again, acknowledging the teams and the agencies that make our work happen. Again, I wanted to thank everybody for the opportunity to be here today, and look forward to the discussion. Thanks again.
Thank you very much, Dean. Perhaps while the questions are coming into the Q&A box from the audience, I can start with a couple of questions. First, to you, Dean. I think many people, and including myself, have this very vague idea of what artificial intelligence is. So is it a specific type, like specific mathematical approach or computational analysis, or is it the application of that to a particular problem?
Great question. I think those are all good ways to look at it. And I think a very foundational way to describe is, you know, taking potentially less amounts of information, feeding it into a mathematical-like approach to make predictions or clinical decisions with that information. The thing with AI is, I think predominantly AI right now is being used for diagnostics, particularly for imaging, and taking large amounts of imaging data and feeding it into algorithms to see if we can make more accurate assessments faster. But I think one of the very valuable areas of AI that are being explored less of is interventional AI. But I also think it's going up against one of the most traditional aspects of medicine, which is selecting treatments for patients. Traditionally, mechanism of action, you pick a drug. Maybe you pick a couple drugs and you give them together. But that approach, while established and effective, is a very different question than optimization, right? If you take two pathways, you put two drugs that inhibit each, you put them together, probably you'll get a pretty decent outcome. But at the same time, success rates are very low when we develop combinations that way. So what's the problem, right? And so optimization in our context involves being agnostic to the mechanism, not ignoring it. We look at it after the fact, but saying, look, I've got 10 drugs and here's a disease model. What drugs do I put together to give the best possible outcome that I can get? And in lieu of using mechanism of action and maybe finding a better outcome, why don't we just black box it, optimize it first, and then go backwards and find out, how did I get this much better outcome? Okay. And when that happens, the answers are astonishing, right? One quick example, we did this, our technology was used for tuberculosis to develop novel combos. And through prospective experiments, the standard of care ranked really, really low, by the way, on this list. It did show up, but ultimately a clinically relevant combination that was been validated in human that cut treatment time virtually in half was realized in orders of magnitude reduction in drug development costs, years faster in clinic, right? There is a top-down approach that AI can be leveraged with to really realize better outcomes. And I would say, to get back to your question, AI indeed is in a generalized form using information to make better outcomes or to predict better outcomes. But AI itself exists in many permutations, and an area that I think deserves more attention is the area of interventional AI.
So a bit of a follow-up question. So there's one from Stacey Zhu, and I had a related question about the experimental validation that you mentioned.
Yes.
And the question from Stacey that is somewhat related is, for the drug combination, will it help selection taking into account of how the human body metabolism or cells will react to the components or the combined therapeutics?
It's a great question. So first off, with toxicity, we always do a counterbalance, right? We don't want to optimize. We don't want to create the most efficacious combination that's also highly lethal, right? And so we certainly do consider things like tox, and whatever we choose to optimize off of is what we work with the clinical team to decide on. And in this case, it would be an index as an example, where you can optimize efficacy and minimize tox against relative platforms. One thing that's got to be considered, though, is certainly our optimization is agnostic to the disease. We can do this for cancer, for COVID. It's agnostic to mechanism. It's agnostic to the drugs. We can have someone mail us 10 drugs, label them 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, and at the very end of the experiment, we will know the rankings. But at the same time, it's agnostic to disease model, in that if some drugs really are not testable in vitro, right, you really need an in vivo model, we can directly apply this to in vivo. We'll just have to reduce the drug count a little bit. But we apply this preclinically to re-optimize our combos. Again, for drugs that require being metabolized in order to function, we don't test those in vitro, right? In many cases, we'll just skip straight to preclinical. In even other cases, we've gone straight to clinical, right? In examples where clinicians say, look, here's a patient, they're definitely getting these 2 drugs. Can you use your quadratic approach to optimize the dosing of these 2 drugs to enhance efficacy? We just go straight to human for that.
Okay. So a question for Linfa. The serological tests that you're talking about give insight into infection history. So, and you mentioned the case of the use in contact tracing. Yeah. But how does this fit into, or does something like CPAS fit into clinical decision-making or public health policy and vaccine studies?
Yeah, so obviously it will play a role, but I think that still very important right now waiting for is the science, right? The basic science, we can measure now neutralizing antibodies, the level of the neutralizing antibodies. But the big unknown right now is what level of the antibody you need then can provide protection. So very practically, for example, this immunity passport, for example, we are not allowed to travel right now because of potential of transmission. But for those people who have been infected, recovered, why they're not allowed to travel? Okay. The reason is that we cannot assess the risk of a so-called second infection. So once we have this data in the future to say, if on CPAS you have 50% above, then you're safe, and the government can issue you an immunity passport to travel. So that's kind of the dream scenario. The more close to home right now is that, even CHOP used the Regeneron monoclonal antibody, and we also talk about convalescent antibody. All these are done in blind. Basically, we just give a combination and we throw, the sink, at the table, basically. And but we find with limited these cases, because the neutralizing antibody, the kinetics is very different from person to person. So we have examples of patients get infected, display a symptom, 2 days later, already have very high level of neutralizing antibodies. But if that person develop into severe disease, and if you give monoclonal or convalescent antibody, it's not going to work. Whether it's going to have a negative impact, we don't know. So that's the second scenario. This antibody can really make, clinical decision. To me, the most exciting or the most important area, I think, is that, we think within the next 3-6 months, there will be vaccine and there will be mass vaccination. From the infection, we can tell not everybody get infected with neutralizing antibody. Even if they develop, the level is different. Even when they have the levels the same, the waning speed is different, okay? And you can imagine vaccination will be the same. So then we come back to scale one, is when you're vaccinated, are you protected from infection? And we said, maybe not. You need to check your neutralizing antibody. So I think I just gave you 3 of these examples that the CPAS or neutralizing antibody determination will play a role in make a clinical decision or even public health policy. Yeah.
Okay. And a question perhaps for both of you, and Linfa, if you could address first, and then Dean, if you want to chip in from your area. But just in terms of, so you briefly went through the response in Singapore, and of course there's been a global response. But in terms of the tools that we have now, are there better alternatives that we should be thinking about that haven't been developed so far? And what tools, if you wanted a perfect pandemic response, what tools would you like to have in your toolbox?
Oh, I think in terms of perfect pandemic response, we have many, many dream tools we don't have. The first one is, even for CPAS right now, it's a 1-hour test, but still you need experience lab-based. So what you need is really something you can deploy and a few tests, but with the same specificity sensitivity. So that's, we have so many lateral flow pregnancy type of tests, but they're just not accurate, they're just not specific. So there's a gap, okay? The second, more importantly, I say, I'm old-fashioned, I think serology is important. That's kind of a half joking. It is important because, you are the expert in next generation sequencing, but we know that the virus does not stay in the body for long, whether it's human or bats or, pangolin. So if we really want to do the pandemic preparedness, there's a good analogy people using is just after 9/11, the US, spend lots of money, basically monitor this chatting, digital signals, whether it's email, WhatsApp, phone, and then you can detect risk signals from there. And the virus are chatting too. The virus spill over to different animals, and the best way to monitor is their antibody. So I think that's a dream too, we're trying to working on, but it's difficult. So currently, all the serology tests we're having is what we ask the question, do I have SARS-CoV-2? You cannot do a serology, what we call discovery driven, say, what do I have? The NGS, next generation sequencing, has achieved that. So we can do that at the molecular level, but we're still not there yet. So I'm, promoting this concept of NGS for next generation serology. Wish me luck. Thank you.
And Dean, same question.
Yeah, so that was really cool, from our end, from the therapeutic side, one thing we've learned is a lot of the questions that we get. We're used to dealing mostly with oncology patients, where you quickly take a biopsy, you optimize regimens, pick a regimen for a patient, and we often pick top-ranked regimens, right, to optimally combat an aggressive cancer, for example. But in this particular case, what we've seen is the clinical communities that have reached out from the various countries for some of this CD clinical decision support information on regimen selection. Sometimes they aim for lower ranked regimens due to drug accessibility, for example. And when that happens, that is making us relook our approach to see if we can better interrogate drug interactions, including on the lower ranked combination spectrum, because maybe this combination, while not optimally effective, will certainly be at least as good as some of the better options out there, while also being accessible, right? And so when we think moving forward, when things first started out, and patients were just being given all kinds of combinations. And I remember some of the earliest headlines were HIV combination cures South Korean patient of COVID, but they didn't really know, right? They give it to 2 people and then they got better, and they could have potentially gotten Diet Coke and gotten better. You'll never know, right? I think that when time is limited, we can do better than just try random combinations and then make assessments off of that. I think that once viruses are isolated, if we rationally run them through an optimization exercise with real experiments, we'll not only be able to try to find regimens that work better, but we'll also maybe find regimens that really should not be tried, right? Regimens that should be avoided, because with patients only have that much time, right, before things potentially get worse. And just blanketing all kinds of random regimens. We're 8, 9, 10 months out, and there are still trials being run on regimens that are probably not going to pan out, right? And I think that if we can decipher that much earlier, this could play a very supporting role in the much bigger picture of diagnostics, tracking, et cetera, et cetera.
Yes, thank you. Yeah, if you pay attention to newspaper headlines, then almost all diseases have been cured at least 3 or 4 times over the last decade.
That's right. That's right.
So we have a question in the Q&A box from Faisal Mustafa, who is asking or saying that they've not heard of interventional AI before, usually in the context of predictive AI, and if you could, any use case that you could perhaps share.
Sure, I'll just give a quick example. So again, we had a patient who had an advanced cancer, and the patient was responding to this investigational therapy, 2 drugs, but was super toxic. The patient was going to quit treatment. And so what we did was we asked for the data, only that patient's data. And with this minimal amount of drug and dose data, we were able to develop a specific quadratic profile just for this patient. And what that did was it told us if you drop the dose 50% of one of the drugs in this regimen, you will actually increase efficacy, right? And this was based off of prospective clinical data used to manage only this patient for treatment purposes. Again, a lot of AI is taking a lot of pre-existing data, feeding it into an algorithm and hoping that this body of data can then be applied to the individual. But in our particular case, if you really want to optimize, a lot of the data really does not exist. I'm not going to take 10 million patients worth of data and then treat the next guy with it. I will actually prospectively calibrate each person, or even take some population-wide experiments to run to actually craft an intervention that is optimized through experimentation versus old data and prediction. And the final thing is interventional, in the sense that, again, a bulk of AI that's out there is used for diagnostics, which is very cool, by the way. We actually work with a lot of the teams that do this. But I think that for treatment selection, treatment optimization, treatment development, there's a big role for AI. And on one of the WHO committees I'm on, which is AI for Health, we are now moving beyond the common terminology of data cleaning, data processing, data this, but thinking, hey, there's different ways AI can be used. And sometimes the data really isn't there. We actually have to prospectively acquire it for interventional purposes, and the future is exciting.
So, somewhat related question, and given that we're the Global Health Institute, is how practical is the use of AI in developing nations? So what are the particular challenges in implementation?
That's a great question. That's a question we're actually addressing in that committee right now, resource limited settings. And I think this is an area where AI will be able to make an impact in resource limited settings once the field progresses to the point where there's this core understanding that it's not the amount of data that we have, it's how we acquire the data. And there are scenarios where you need less patients and less data to truly optimize. But in order to make that happen, how trials are run and the designs of those trials will need a reevaluation. And we're getting there, right? And even before COVID, I think there was an argument to be made where if you design trials properly, you can avoid the error, avoid the substantial resources needed, and actually be even more sure if an intervention works or not. But in order to really make that widespread and more applicable to resource limited settings, there has to be a rethink about how interventions are trialed and developed and beyond. But again, it's not always how much data. It's not always about having a huge data infrastructure. It's really how the data is acquired.
Thank you. And Linfa, I wanted to follow up on, you mentioned a couple of studies at the end. So one of them was using CPAS throughout Southeast Asia, so areas where there's high numbers of bat populations, etc., to look for the source. So I'm not sure I understood correctly. So is the idea that that there's been a related coronavirus circulating in humans or in an intermediate host in that region?
We don't know. The hypothesis, it could be both, right? So, Antonia's work is really kind of, it's a very important discovery, but imagine that, 50% of the healthy donors, I mean, it's a small number for the individual in Singapore. When you check their T cells, you find that they have cross-reactive T cells to SARS-CoV-2. And the peptide says that it's not a seasonal coronavirus, either SARS-1 or SARS-2 if you have been exposed, and then you will have this kind of T cells reactivity. Obviously, Singapore was hit with SARS pretty bad, but it's 300 patients, right? So 200 something. So the hypothesis is that either there's an unknown coronavirus has spilled over, caused mild disease or no disease, and we don't know, but it's related to SARS-1 or SARS-2. So that's the T cell work. In terms of the antibody, we have this CPAS, which detects neutralizing antibodies. So if we detect any animal or humans in our region have that antibody, but there's no history of SARS or COVID-19, especially that's why we're interested in archive zeros, means the zero, collect for human especially, is before December 2019. And then if we can find these, either in human or in animals, and if there is a localized region has a high prevalence, then that gives us some kind of a hint. Maybe we should intensify not only serological investigation, but also molecular investigation. So serology is important, but it has to go hand in hand with the molecular. I found that serology is much more sensitive because the antibody can last years, whereas the virus maybe last 2 weeks. So this is the whole justification, and WHO think it's a low-hanging fruit using archive sera or fresh example sera, and to do CPAS type testing, basically start with Asia, but all over the world. Because you have seen this, reporting of in the sewage in Spain, they detect the SARS-CoV-2, PCR positive in March 2019. Most people try to ignore that as contamination, but there is a possibility of rare spear event which did not lead to COVID-like pandemic, but actually caused by a close-related virus. Yeah, so that's possible. Yeah.
Okay. Thank you. So Dean, I had a question about the different combinations that you test. So presumably you talk about aiming for an optimal combination, but as we know in a pandemic, there's very uneven distribution of resources. I was listening to a talk from a clinician in Hong Kong and saying that they just do not have access to some of the drugs that you were talking about. So I assume that the approach that you're looking at is quite flexible in terms of drug availability and, assessing even the utility of suboptimal combinations based on availability.
That's a great question. Yeah, actually, so again, one of the biggest differentiators with our approach from traditional drug development is you go in actually knowing what combination you're testing. If somebody says, I think this drug and this drug is the best, I'm going to go test it. In our case, we don't know the answer until the experiment's done, right? And so we put the 12 drugs in, and we picked those 12 drugs in collaboration with various clinicians from various parts of the world to try to forecast where we think, and one of our first discussions was around March with the former CDC epidemic intelligence officer to forecast, hey, we didn't want to make a US-centric set of drug combinations, right? We wanted to try to understand where they thought supply chain disruption would occur and which drugs may emerge as leading candidates. You know, some jurisdictions liked favipiravir, some of them like this, some of them like that. We use that to pick our drugs, but once it's done, we don't just yield one answer of what the optimum is. We actually get a full list of ranked combinations from best to worst, ranked based on this efficacy window, efficacy safety window, whatnot. Now, once that's done, we have a database, and we query that database where if a clinician says, look, I don't like giving 3 drug combinations. They're always going to be too toxic in my book. So can you find me the top 2 drug combination you came out with, but that does not contain this drug because I don't have it? Or give me the top 2 drug combination that doesn't have this drug because I know this patient has a comorbidity and they can't take this drug. So this one experiment yields a ton of answers at once that can be queried and adjusted based upon various factors. The only thing is, what we do now is we're going to run new experiments with newer sets of drugs and just keep adding to this what will be a publicly available database. Again, the most prevalent question we get is, this is in vitro, okay? How applicable is this clinically? The real thing there is, again, I would say our best foot forward is certainly finding number 1 out of 1 trillion, number 1 out of 530,000. In some cases, we'll have to settle for number 400, which is potentially still pretty good. At the end of the day, if given the opportunity, you're still going to want to run the preclinical optimization and certainly clinical validation. What I will say is, time and time again, in vitro, ex vivo, the top ranked combinations have held true clinically. And the reason for that is because you're finding number 1 out of 500,000. You're not kind of going in with, this is going to be inhibited, that's going to be inhibited, let's just try to carry this all the way through.
So I think a lot of those decisions are being made in the clinic anyway, particularly during a pandemic where you know, the clinicians, they know the past usefulness of the virus, but in particular with relation to SARS and so forth. And try new combinations.
They just kind of try them out, right? But in this case, we can really leverage some really unexpected interactions.
Yeah. So we're running out of time. So I thought very quickly for closing points. So the vaccine or vaccines are going to be our great savior. So very briefly for both of you, Dean, is AI applicable to vaccine research? And Linfa, when are we going to get a vaccine?
Quickly, I think it's possible, but I'm actually really interested so that being said, it was wonderful being here today. I think it's certainly possible with trial design, in particular for vaccines. I think the design of the trial is possible, but I'm actually really excited and interested in Linfa's take on what's next.
Well, if you want me to predict, I think the next 6 to 8 weeks, I think we will have a ranking of who are the front runners. And then I think as long as they're safe, because the efficacy is the hard one, right? You know, is it going to be 50%, 30%? I think, you know, governments are so desperate. So as long as we have a safe vaccine and the efficacy is 30 or 50% or above, I think we will have a mass vaccination. But then you have the supply chain, and then you have the, you know, a socio-economic sort of consideration. So mass vaccination, I think the earliest will be first quarter of 2021. So we're still not allowed to travel until maybe middle of next year. You know? That's my prediction.
Fingers crossed for travel bubbles. Okay, thank you both very much, Professor Ho and Professor Wang, for your contributions this evening. And I'll sign off, and I believe Amina has some closing remarks. Thank you both.
Well,
Okay,
I don't really have many closing remarks, but just to say thank you to both the panelists. But I will make one closing remark to reveal my ignorance, because I always associated AI and big data together. So I'm going to go and read more about it. That's for sure, because Dean, you really kind of opened it up in a very different way. So I hope the audience kind of enjoyed that also. I certainly did. I think in this little presentation, I learned a lot, and I want to go and read more about it because it really sounds fascinating. And it really takes me away from the way I conceptualized AI. So this is, quite different and obviously, applicable in our day and age. And then for Linfa's closing remark about the vaccines, I do want to just let everyone know that this is the final webinar series in this healthcare innovations topic. But our next topic is actually titled Beyond Science. So figuring the science has been worked out. How are we going to access the vaccines and the drugs that hopefully we'll have in the 6 weeks or to 6 months, whenever it comes out? But we'll be looking at, issues of legal issues, financial issues, distributional issues in our next webinar. So please join us there. Gavin, thank you so much for moderating the session and bringing out important topics to discuss. And hope to see you all again in this venue again.
Okay.
Thank you. Good
Thank
night,
you so
everyone.
much. Thank you so much.
Bye.
Take care.